I'm Michelle. I'm from the Canadian Magor Lab, and today I'll be talking about the duck NLRP3 inflammasome is not activated by nigericin. And so we're interested in the duck because it is the natural reservoir host of the influenza virus. It harbors almost all known strains with little to no detrimental side effects. And this is unlike other hosts like humans or Galliform poultry. And so one of the immune pathways activated by the influenza virus is the NLRP3 inflammasome. And so the NLRP3 inflammasome has three components, NLRP3, ASC, and caspase-1. Activation of the NLRP3 inflammasome causes the release of pro-inflammatory cytokines, namely IL-1 beta and IL-18, and they will induce an inflammatory cascade. So in mammalian hosts infected with a high path strain of the influenza virus, there's a dysregulated overactivation of the NLRP3 inflammasome. And so this leads to cytokine storms and hyperinflammation occurring in the area of infection, in this case, the lungs. And this is correlated with worse host outcomes and potentially leading to death. And so the duck NLRP3 inflammasome has yet to be characterized. And since ducks don't have the same severe outcomes as other hosts, we thought that the duck NLRP3 inflammasome would function or respond differently. And so the first step was to generate expression constructs containing these different components of the NLRP3 inflammasome. And so using bioinformatics and transcriptomics, I was able to find sequences of these components, and then I was able to clone duck NLRP3, duck caspase-1, duck IL-1 beta, and attach different epitope tags to them. However, I was unable to locate duck ASC in the genome or the transcriptome. And so this is a major difference between the duck NLRP3 inflammasome and mammalian counterparts. And so using these constructs, I performed some co-transfection experiments and took a look at them through confocal microscopy. And so these are all done in DF1 cells, which is a line of immortalized chicken embryonic fibroblasts. And we use these because they're a little bit easier to transfect than the duck embryonic fibroblasts we have available in the lab. And so here in this first column, you can see the caspase-1 in red, NLRP3 is here in green, the hooked stain showing the nuclei of the cell here in blue, and then the merge. And so caspase-1 forms these tight little punctate structures within the cytoplasm of the cell, while NLRP3 forms this more diffuse in the cytoplasm of the cell. When you merge them together, while they are both localized to the cytoplasm of the cell, there's very little actual co-localization between the two of them. And so the other thing I wanted to look at was the duck NLRP3 inflammasome activation. And so I took some duck embryonic fibroblasts, treated them with poly-IC or poly-IC and nigericin. Nigericin is a known NLRP3 inflammasome agonist, and poly-IC is it mimics a viral infection. And so the NLRP3 inflammasome, you have to prime before you activate. And so the poly-IC is that priming step, and nigericin is the activating step. And so I looked at the gene expression of these different genes through qPCR. So I looked at NLRP3, IL-1 beta, and IL-18, as well as IFIT5 as a positive control. And so we can see that IFIT5 here is upregulated in these cells, indicating that cells do respond to treatment. However, when we look at the components of the NLRP3 inflammasome, we see that NLRP3, IL-1 beta, and IL-18, all of them show little to no upregulation, indicating that nigericin does not induce any activation. That's just to sum it all up here. So the duck NLRP3 inflammasome does appear to respond differently compared to its mammalian counterparts. It also does not appear to have ASC, which is one of the components of the NLRP3 inflammasome. And there's no induction after treatment with nigericin, which you see in mammalian cells. There's a similar situation in other birds. The NLRP3 inflammasome and other species do behave similarly. However, unlike ducks, influenza virus still causes severe symptoms in these other species like chickens and Galliform poultry. And so the NLRP3 inflammasome remains unexplored in chickens in this context. And so I'd just like to thank my supervisor, Dr. Kath Magor, as well as other members of the Magor Lab, and my funding provided by the Department of Biological Sciences and CIHR. And I'll be happy to take any questions.
Thank you, Michelle. Great. So I remind everyone that if you have questions, please type them into the Q&A box and I'll be reading them off to our presenters. While we wait, I have a question. So I monitor issues related to avian influenza, et cetera. But I was just wondering, you mentioned, so it's unexplored in chickens, and obviously birds still get influenza. I was wondering if there's been any work done in swine, as pigs are also known to get influenza, and there's evidence that it could be passed that way. So can you, I'm just putting it out there, can you comment on that at all, and how swine might play into this, if at all? Thanks.
Yeah, there is some research done on the NLRP3 inflammasome in swine and just like other mammals, because there's just a lot more reagents available to study mammals. And so the NLRP3 inflammasome in swine is a lot like the sort of classical ones in mice and humans. So you do see the activation and like a similar response as like humans or mice would when they're infected.
Fantastic, thanks. Question, so how would this research help for the development of a vaccine?
So I think that this is less of a vaccine route and more of like a therapeutic treatment post infection. Sort of like, how could you still clear the virus but not get that terrible inflammation that would end up killing the bird or what other organism you're looking at. So it's not quite like a vaccine in that sense, but more of just like, how do you deal with it after you've caught the virus?
Okay, and we do have another question. They're going to make you work here. So are
Okay.
there other inflammasomes involved in addition to NLRP3 in chickens and ducks?
There are quite a few other inflammasomes just in general, because just how nasty like the virus is, you get a ton of different inflammasomes being activated because you get all this inflammation. But the main one is the NLRP3 inflammasome, and that's like the most well-studied one that people have linked to the influenza virus. But yeah, there's loads of other inflammasomes at play.
Excellent.